I just want to thank all of you for being here and for your support, and I'm trying not to tear up what Gary and Stuart said. So wetlands, people or scientists often say that you will know a wetland when you see it. And that's because they're really, really challenging to define. They are this interface ecosystem between terrestrial systems and deep water. And so what you get is a system that has intermediate properties in hydrology, or the nature and flow of water. They're also highly productive systems. So if you combine those fluctuating water levels with the high productivity, what you end up with is a biogeochemical hotspot where lots of processes occur. One of those processes is decomposition, or the recycling of carbon. So if you want to imagine that you're in a really productive wetland where there's lots of plants, right? What happens to those plants when they decompose and die? Well, essentially, that carbon is recycled, so it can be used by other organisms. And so this right here shows you, these were plants that were placed in wetlands where I work. And essentially you see decomposition right here on the y-axis, and the x is, again, these different types of vegetation. And it's clear that these different types of vegetation decompose at different rates. But what's really, really interesting here is if you look at these little C and F labels, that stands for a coarse mesh bag and then a fine mesh bag. So the coarse mesh bag had that vegetation in it, and it allowed invertebrates to come through and help with the decomposition, whereas those fine mesh bags only allowed microbial organisms. And as you can see here, there's really no difference between these coarse and fine mesh bags. And that just goes to show you that microbes are doing the majority of the work of decomposition in wetlands. And so I didn't set out to study microbial organisms, but as I started getting involved in looking at wetlands, it turned out that they were the ones doing all the hard work. And so that's going to be one of the themes throughout my dissertation. So if we take and look at decomposition at more of the cellular or chemical level, you can imagine these complex molecules, right, from those plants or organic matter, and those get broken down into simpler compounds, think something like sugar. And microbial organisms use those simpler compounds and the electrons from those simpler compounds to generate energy or to make their living. So when I talk about electrons, you can just think of it as a type of currency, right? Or again, making a living. And so what happens is when this organic matter donates electrons, there has to be something that accepts those electrons. And in our case, in the cells in our body, as well as many microbial organisms, they use oxygen to make their living. That's what accepts those electrons. And so it's the reason why we breathe in oxygen, right, and we process those simple sugars, and we exhale and we release carbon dioxide and water as our waste products. Turns out microbial organisms have a lot of creative different ways of making their living. They can use nitrate right here, iron, sulfate, and carbon all as electron acceptors, things that we cannot do. They're really fascinating. But it turns out that not all electron acceptors were created equal. And so I've created this diagram here, this tower of power, to essentially illustrate that up the top here, this is oxygen, that's the most energetically profitable electron acceptor, okay? So if we were gonna take the ride and we're gonna drop it from the top, right, it's gonna pick up a lot of speed and release energy as we get to the bottom. Whereas if we drop it lower down on the tower of power, we're not gonna pick up as much energy, okay? So after oxygen, we have nitrate, which is in line, and then after that, iron, and then here, sulfate, we're getting towards the bottom here, and then when you're using carbon as your electron acceptor, you're just getting a little bit desperate, okay? And so one thing I want you to think about is that if the resources in an environment are scarce, what's gonna happen is these different microbial organisms are gonna compete for those resources. And ones at the top of the tower of power are likely to outcompete the ones at the bottom, okay? You can think of it as a microbial warfare of sorts. So another thing I want to tell you about the bottom of the tower is if you happen to use carbon as your electron acceptor, and carbon that can be in the form of organic matter or even carbon dioxide, the waste product is methane. And that's going to be really important because I'll focus on a lot of my talk on that particular process. So another theme in my dissertation is that I'm integrating the disciplines of physics, chemistry, and biology to look at how pond ecosystems function. Another theme is that I'll be studying this across multiple scales. So we did experiments in bottles in the lab. We also did some at a pond level, and then we even expanded it out to look at a landscape level. And then lastly, the theme that unites my research is where it takes place, or the study area. And so the Copper River Delta is located in south central Alaska. The Copper River itself is the 8th largest river in the United States. It drains an area about the size of West Virginia, okay? So it's a pretty large-scale landscape. The Copper River Delta is also influenced by many different glaciers in the near vicinity, around 7, depending on which ones you're counting. And so what's pretty neat here is if you check out this photo from NASA, right, you're seeing the copper, right, which is fed by this glacial melt during the spring and summer. And that glacial melt brings with it a lot of sediment that gets dumped into the Gulf of Alaska. You can see it from space. It is so much, right? And we call that glacial flour or glacial sediment. And what's really cool is it turns the river this kind of milky color. You can also see it, not only see it, but hear it. If you're in a boat, it will kind of swish against the side of the boat. And so it really characterizes these systems in a really interesting way. And so if you look at this picture right here, this is the famous Childs Glacier of the area. And then this right here is the infamous $1 Million Bridge. Okay, this is what happened to the $1 Million Bridge after the Great Alaska Earthquake of 1964. It's a 9.2 magnitude earthquake, the second largest ever recorded in the world's history. So huge earthquake, ruined the $1 Million Bridge, and shifted the delta upwards several meters in places. So it gave rise to this really nice succession of mudflats to brackish marsh, uplifted ponds, and then glacial outwash ponds closer to the mountains. For the majority of my dissertation, I'm going to be focusing on the glacial outwash ponds and the uplift systems. But we'll dig down and talk a little bit about the brackish marsh just for fun at the end. Bonus. So, the glacial outwash systems, as I mentioned, are close to the glaciers. They're near the mountains. That's not going to stay there. So essentially what you see here is the glacial outwash ponds tend to have a lot of glacial sediment that brings iron to these systems. And they also have lower dissolved oxygen levels, especially when you get towards the bottom of these ponds. So here's a picture of me trekking out to one of these outwash ponds. And what you see is this trail of rust, or iron oxides. And what's really fascinating about that is actually bacterial poop. So iron oxidizing bacteria, which are shown in this picture here, they're actually so small that you can barely see them. They're actually dragging along these squiggles here. That's their metabolic waste. So I don't know, only bacteria could, essentially drag that much along with them. But needless to say, what's really cool about these iron oxides and these bacteria in the system is iron oxides, because of those low dissolved oxygen levels, mean that iron is actually a pretty profitable electron acceptor. It's a good way to make a living in the system. Whereas if we get closer to the ocean and these uplifted ponds, you get these really productive systems where plants are photosynthesizing, they're producing oxygen, and because of that productivity, right, and those high oxygen levels, you might find them higher up on the Tower of Power than you'd find those glacial outwash systems. And so essentially this Tower of Power is going to help illustrate where some of these ecosystems fall, and therefore what processes are going to take place in them. So for the entirety of my talk today, we're going to take multiple trips up and down the Tower of Power here. We're going to start up and talk a lot about processes that affect oxygen, specifically decomposition, as well as photosynthesis at the top at a more landscape level. Then we'll drop down and talk about nitrates' effects on biofilms. And then at the very end, we're going to mainly spend a lot of time at the bottom, where we're going to discuss the methane cycle. But just for funsies, we'll hop back up and talk about sulfate as well, just for your encore ride there. So let's talk about chapter 1. And essentially we wanted to look at this really diverse, heterogeneous landscape And so we sampled 15 ponds throughout the Copper River Delta. These green triangles are glacial outwash systems. The yellow ones are uplifted ponds. And so what we did is we essentially wanted to look at how the landscape from glacier to ocean here influences chemistry and processes. And so we used glacial flow path length was essentially the glaciers up here, you can see in the blue. You can imagine the distance it takes for this glacier melt to sort of flow over land to this pond right here. Rotate me, and that would be the glacial flow path length, or kind of an indicator of glacial influence. Whereas distance from the ocean was our indicator of oceanic influence. And so our first question was to ask about how does the chemistry vary across this really unique landscape? And we measured lots of things. Don't worry, I'm not going to tell you about all of them, but I'm going to tell you about the most interesting things that we found. And so I want to get you oriented on this plot here. If we look at the x-axis of both of these, this is glacial flow path length. So at 0 here, this means we're really close to a glacier. We're like right on top of it. As we get farther away here, that means the pond is farther away from the glacier. And so what we saw here is that iron tended to decline precipitously as we get farther away from the glaciers. And the same thing with phosphorus here. Phosphorus also tended to decline farther away from the glaciers. And we think that has something to do with how the glacial weathering is occurring. So that physical abrasion of the geology in that system is releasing iron and phosphorus. So now let's turn our heads, so to speak, and think about some of those patterns from the ocean. Okay, on the x-axis here, you have distance from the ocean, very close to the ocean, farther away from the ocean. And the first pattern we saw is that dissolved organic carbon, or DOC, tended to be really high closer to the ocean. Dissolved organic carbon comes from plants and algae sometimes, so we thought that for whatever reason, there was either more productivity in the terrestrial or aquatic landscape leading to these patterns. We also saw higher ammonium levels, which is just a form of nitrogen, towards the ocean. And essentially, we're not exactly sure what causes that, but we have a couple of different hypotheses. And one could be that there is just higher deposition of nitrogen from the atmosphere in those coastal areas. And another one is that sometimes there are high nitrogen levels in some of the marine sediments, another possibility, as well as it's possible that there are higher nitrogen fixation rates by organisms like cyanobacteria closer to the ocean. So let's actually look at how that chemistry influences ecosystem function. And so when we're talking about ecosystem function, we're talking about processes. And so when we're measuring decomposition, one way to do that is take a cotton strip, which is supposed to approximate like a leaf, and essentially you put it in these litter bags, and what we do is we incubate them in the ponds over time and then rip them in half to essentially look at that change in tensile strength. So we're looking at how fast those cotton strips decompose through time. Another thing we also measured was ecosystem metabolism. And you can kind of think of this as the heartbeat of an ecosystem. And so ecosystem metabolism, there are two parts. There's GPP, gross primary productivity, and that essentially is the productivity of the plants and the algae in those systems. Whereas if you're thinking about ecosystem respiration, that removes oxygen from the system, whereas GPP adds it. And so ecosystem respiration is like you and me respiring, right? Or consumers or microbial organisms. And so essentially, both of those processes are going to be affecting our oxygen levels and therefore how far up on that tower of power we are. So this was our decomposition rate that we measured with those cotton strips I was telling you about. And as you can see, the systems that had higher amounts of phosphorus tended to have higher decomposition rates as well. And what's interesting about this is that previous study that I showed you where the different types of vegetation varied in decomposition rate. Well, it just so turns out that these ones over here that decomposed at faster rates had higher amounts of phosphorus in the tissue. So you can kind of think that higher amounts of phosphorus in the tissue or in the water column where we measured it tended to lead to higher decomposition. And that may just be because those microbial organisms are limited by the amount of phosphorus that is there. So on the right here, we have ecosystem respiration, or again, that's all that consumer microbial activity. And we saw that ponds that had higher iron levels also had higher consumer activity. And the cool thing about this is that if we remember, right, near the glacier, we had higher phosphorus and higher iron levels, and therefore also higher consumer activity. Now let's go ahead and look at our plant and algal productivity. And again, our GPP here and our dissolved organic carbon. So in systems that had high plant and algal productivity, they also had high DOC. We think that that DOC was really coming from the plants within the system. A lot of these wetlands have really big macrophytes, like lily pads. Macrophyte's just a word for big plant. And so they have lots of that. So we think that is internally produced and sort of an indicator of productivity. Just as this total nitrogen was higher in those ponds as well. And so these ponds tend to have pretty low nitrogen levels, so it's possible that the plants are really limited by that nitrogen. They need that to photosynthesize. So to sum this up, what we found is that closer to the glaciers, we had higher iron and phosphorus, and that led to more consumer microbial activity. And what's cool about that is that we tended to see that the physics, right, that glacial weathering really shaped the chemistry of these systems, which then influenced the biological processes. Whereas when we got closer to the ocean, we saw higher dissolved organic carbon, more nitrogen, and we actually think that a lot of those patterns were actually driven by the biology instead of the other way around. So it just goes to show you that the physics as well as the biology can really shape the chemistry in these systems. So now we're going to drop down on the tower a little bit, and we're going to talk about nitrate and how it affects biofilms in these systems. So imagine you're in a stream, you pick up a rock, super slimy. Biofilm is that matrix of algae, bacteria, fungi that's just growing on that rock, right? And so it turns out when we are in aquatic ecosystems, we often think about whether nitrogen or phosphorus is going to limit those biofilms. You can kind of think of those nutrients as like a vitamin or something for the biofilms. And so we wanted to see, well, we have low nitrogen and phosphorus in the Copper River Delta wetlands. Will our biofilms be limited? And so to test that, we use this technique called nutrient diffusing substrates. And it's essentially a cup that we fill with an agar or jelly-like material. And then we put a glass disc on top. That serves as that sort of substrate for colonization, right? Think of that rock, right, where the biofilm can grow on it. And then we will add nitrogen, phosphorus, or both, and then look at the growth of our biofilms. And so we did this across 15 ponds, and we measured chlorophyll A. Chlorophyll A is a good indicator of our algal abundance. The algae use that pigment to photosynthesize, right? And because when we pulled out these biofilms, they tended to have more than just green algae on them, we wanted to make sure that we were capturing the entire community. So we also measured what we call ash-free dry mass. And it's just because organic matter burns, that's a good way of measuring the total amount of biofilm, okay? So let's look at one of these plots from one of my ponds. And here, this is our chlorophyll A, or our algal abundance, right? And we have our control, that's where no nutrients were added. We have our nitrogen in red, our phosphorus in yellow, and our nitrogen and phosphorus treatment in blue here. And essentially, I'm going to be talking about all of these different nutrient treatments relative to the control. And to do that, we use what's called a response ratio. So in the case of phosphorus, right, we know that there's more chlorophyll or algal abundance relative to the control. That ratio is greater than 1. We know the nutrient is limiting. Whereas if we look at something like this nitrogen here, that's clearly less algal abundance than the control, so that ratio is less than 1. That nutrient is inhibitory. And so let's look at the results combined across all 15 ponds. And so this line right here is where the control growth was at the 1 here. So what we saw is that when we added phosphorus, we actually doubled the amount of algal abundance on those discs. Whereas if we added nitrogen, we actually didn't really see that much of a change from the control. If anything, we saw slightly less algal abundance. But if we look at the total biofilm biomass, so everything that's on that disk, not just the algae, what we saw is that really adding no type of nutrient tended to increase that amount relative to the control, right? These are all less than 1 here. So phosphorus had the most similar biomass relative to the control, but what's really striking here is that when we added nitrogen, nitrate in particular, there was like a reduction in biofilm biomass by over 60%. You saw less than half the growth, which was really, really striking. And this nitrogen plus phosphorus was sort of intermediate. You might think that that's due to some of the boost in the algal abundance that we saw earlier on the previous slide. But we thought long and hard about this because we did this in 15 ponds across 3 years, and we're always seeing these strong inhibition of the biofilms by nitrogen, which was weird because we know it's low in this system. Why is that an issue? And so we tried a different type of nitrogen. We tried ammonium. And what we found is that ammonium really didn't have the strong effects like the nitrate did. It looked more like this phosphorus here, so it didn't change the amount of biomass. And so that got us thinking, what is it about the nitrate? And if you know where I'm going here, then you are actually farther ahead than I was. But so our next step was actually to dive into the microbial communities that were present on those different discs and see who was there. And so what we did is we extracted DNA from those discs, and we wanted to look at which microbial communities were present. There's often thousands of microbial species, and that's really hard to think about. So what we did is we used this ordination plot to help you think about the data in 2-dimensional space. So each one of these is one of those discs from a different pond. And so the closer those 2 discs are to each other, the more similar those microbial communities. The farther away, like these two, have pretty dissimilar microbial communities. And so if we look here at the control biofilms, remember that they had really thick biofilms, and they tended to have green algae plus this sort of non-sulfur purple bacteria. They had really unique metabolisms and were really, really interesting and thick. Whereas our phosphorus-emitted ones were also closest to the control in terms of the communities. But then when we added our nitrate, that's where we got that drastic reduction in biofilm biomass. And so we ended up with organisms that could either use nitrate as their electron acceptor, so make their living with nitrate, or because how thin those biofilms were, they could also use oxygen as well. So really different communities here, and again, drastic differences in the biofilm biomass, as you can see. Whereas if we looked at our nitrogen phosphorus results, those were kind of intermediate. And this is very similar to what we saw for our total biofilm biomass. So you see those microbial communities really sort of echoing those results. So now onto the million-dollar question. Why might biofilms be inhibited by nitrate? And so one possible reason is nitrate toxicity. So it turns out that we often use nitrates in our cured meats like bacon and other things, and we use that to reduce microbial spoilage. So it's possible that some of these microbes didn't do particularly well in the presence of nitrate. Another thing is, I told you about those shift in those biofilm communities, right? Where we saw that the discs that had the nitrate added to them tended to have organisms at the top of the tower of power, whereas those on the control used some of those more creative electron acceptors and had more creative ways of making their living towards the bottom. Thirdly, it's possible that in these systems there's really low nitrate levels. There's very little geological or nitrate coming in from fertilizers or sewage. And so it's just possible these biofilms aren't adapted to those types of nutrients in comparison to other wetlands that have essentially been receiving nitrate for hundreds of years or at least over a century. And that really primes those communities, whereas ours aren't as likely to respond to it. So let's drop down to the bottom of the power of tower and talk about methane. So methane is a really important greenhouse gas. It traps about 30 times as much heat as carbon dioxide. And if you look here, This is the amount of methane emitted to the atmosphere from wetlands. It's about 60% of all natural sources. And that's actually the same amount of methane that is emitted from fossil fuels over here, rice cultivation, and cow farming combined. So, as a really important greenhouse gas, again, that traps heat, it's really important for us to understand more about the methane cycle in these wetlands. So let's go ahead and dive in and talk about what this cycle looks like in these ponds. So we have methane that's produced in the sediments here by microbial organisms. We call those methanogens, meaning they generate methane. And so that is the source of methane to these systems. And so those methanogens often will produce more methane when there's higher temperatures. It's just higher microbial processing rates. They also tend to do not very well in the presence of oxygen. They just don't tolerate very well. They also need electron donors, right? They need that organic matter to make their living. So all of those factors are really important for influencing that. That methane that then gets produced can be oxidized by methane oxidizing bacteria. And so what they do is they take that methane in the presence of oxygen and they convert it into carbon dioxide. And so both methane and the amount of oxygen are really important for thinking about how much of that processing is occurring. The methane that's not oxidized is often stored temporarily in the water column, and then that can be emitted to the atmosphere via 3 different mechanisms. The first one I'm going to talk about is diffusion. And so you can just think about methane building up in the water column here. If it's higher in the water column relative to the atmosphere, those methane molecules are going to try to become a little bit less crowded, so they'll move to the atmosphere essentially. Another mechanism is what we call bubbling or ebullition. And so what happens is that instead of the methane being dissolved in the water column, it will actually build up in a gaseous form in the sediments. It gets trapped in there, and then it will take a while for it to essentially bubble out and vent to the atmosphere. Our third mechanism is plant-mediated transport. And you can just kind of think of this as the plants in these wetland ecosystems serve as giant straws that vent methane to the atmosphere, okay? And so these emission processes are often more controlled by physical things, such as the depth of the pond as well as the wind speed, and we'll talk about that in a little bit. So I wanted to study the pond methane cycle and determine how predictable these different processes are. And so what we did is I went out and I measured methane production by taking sediment and water from the ponds and then measuring the change in methane concentration through time that these little bottles were producing. And then we also measured methane in the water column as well as the amount that was being emitted to the atmosphere using these flux chambers, or they're just sort of glorified mixing bowls with a pool noodle around them that float on the top of the water that capture that methane that's being emitted to the atmosphere. And so important thing to note is that sometimes we're capturing diffusion, sometimes we're capturing bubbling, and sometimes we're capturing plants, but it's hard to disentangle between those 2 or 3. So then what I did is I took a bunch of these factors that I talked about earlier, right, where I told you how the methane cycle is influenced, and we measured those as well, like temperature, dissolved oxygen, other things. And we essentially put those into statistical linear models so to see how well we could predict these different processes. And so first we measured our methane production, right? This is the actual values that we measured in our ponds using those bottles. And then this is our predicted methane production. And so what you see here, this dotted line here is the 1-to-1 line. So that is where all the points would fall if we predicted methane perfectly. We didn't, obviously, but if you look at that solid line that's generated from our statistical linear model, we actually did a pretty good job approximating that 1-to-1 line. We explained about 54% of the variation, which for such a heterogeneous process is pretty decent. And we used the following factors to do so. So dissolved oxygen, or DO, had a negative effect. Again, remember how I told you oxygen tends to affect methanogens, or those organisms producing the methane, negatively. Also, temperature increased methane production, again, higher microbial processing rates. The microbial communities in those sediments also mattered. So if we looked at the amount of methanogens relative to their competitors, we found that higher levels of that actually tended to produce more methane. And algal abundance also had a positive effect. And remember that the algal abundance is sort of this indicator of the organic matter and the quality of that in the system. And that's been shown to have a positive effect on methane production before. So now let's jump down and think about methane concentration here. And what we see is that we tend to see a lot more physical factors come into play. And so, interestingly enough, methane had a positive, or wind speed had a positive effect on the methane concentration. Usually when we think about deeper systems like in lakes, we tend to think that wind speed is going to increase the amount of methane that's emitted to the atmosphere. So therefore what's left and what we can measure in the water column is going to be lower. But actually in these systems where it's really, really shallow, it's possible that the wind can actually mix the entire pond. So it's actually mixing some of that methane that's produced in the sediments and stirring it up and getting it into the water column. Now, dissolved oxygen and depth both had negative effects on the methane that was in the water column, because both of them tend to increase the amount of oxidation that's occurring. And again, that oxidation is removing it from the water column, so you're not measuring it. Also, we saw that there was a positive effect, but it wasn't quite as important as like wind speed, was methane production. And again, as the source of methane to the water column, right, that makes sense. We were able to explain a little less variation than methane production, about 41%, but still pretty decent. So let's talk about diffusive emissions, right? And what we saw here is that methane concentration had that strong positive effect. Remember, the amount of methane that's in the water column, the more there is, the more likely it's going to diffuse to the atmosphere. Dissolved oxygen had a negative effect. Again, that means that more is oxidized, so it's removed before it has a chance to be emitted to the atmosphere. Wind speed tends to increase it because, again, it's stirring up that water column, getting more of that methane up into the atmosphere. And then methane production, of course, is a source. So here we were able to explain about 45% of the variation. But here's where things get really tough, is when we start to talk about the total emissions. And remember, that's all those emissions coming from diffusion, as well as our bubbling and our plants. And we actually were able to predict less than 1% of the variation. And that might have to do with the physics and how important the physics are in this process. Especially when you think about bubbling, which has sort of a chance-like nature anyways, and so it's just really hard to explain when and where that's occurring. So what we did is I put all of our own measurements of these ponds on this diagram that I was talking about earlier. And we used the amount of methane produced and what was in the water column and removed the diffusion from it. And we were able to calculate oxidation. We didn't measure this directly. But what we found is that about 35 to 98% of the methane that is produced in the sediments is actually oxidized in the water column, which is similar to what other systems have seen, like in lakes, for example. But I think what was really interesting is that if we tried to balance these methane budgets with total emissions, we weren't able to do that. And that's because the total amount of methane being emitted from the system was higher than what we were measuring that was being produced in the sediments. And that was really interesting and pretty curious. And we think it might have to do with the nature of bubbling in the systems. So if you can imagine that methane is being produced in some of those sediments in a gaseous form, right? It takes time for that bubble to build and build and build. And so perhaps that methane that's being produced, it may take months or even years, which is longer than the time scale we were measuring our methane production. So it's just sort of integrating that methane over longer time scales. It takes much longer for that to bubble and reach the atmosphere. The other interesting thing is when we looked at our measurements compared to other systems, our methane production and water column concentrations and diffusive emissions were all pretty modest. But when we looked at our total emissions, which actually spans several orders of magnitude here, right? You see a whole lot of variability. What we found is that on the higher end, our total emissions are much higher than other systems. And I think it just goes to show you that shallow ponds can disproportionately contribute to the methane budget, but they can also be really challenging to predict. So let's think a little bit about how methane can be traced into the food web. And so I told you earlier, right, methane can be produced, it can be oxidized by methane oxidizing bacteria, and then that methane can be incorporated into the food web to invertebrates like this one by when they chomp on that bacteria, okay? And we can actually trace this with stable isotopes of carbon. And really the only thing I need you to know about this slide is that methane is really, really low in the carbon isotopes that we are measuring relative to all the other carbon sources. It sort of just breaks the scale. And so that methane can be traced in the food web with these isotopes, kind of like a dye. You can imagine seeing, right, like a chemical dye. And so what we did is we know that methane has been incorporated into the food web, at least for consumers, but we didn't know as much about what happens when vegetation is decomposing. And so if you can imagine that you are a piece of vegetation, you're decomposing in a pond, right? Microbes are eating you, they're eating your carbon, right? So they're gonna change the amount of heavier carbon and lighter forms of carbon that you contain as they are working and processing. And so if you have microbial organisms that are making methane, and they're making methane from your tissue, this methane tends to be really light, right, in those isotopes, like I showed you earlier. And so it's really, really light. That means you're left with the heavier forms of that isotope. So then you would see an increase in your carbon isotope signature with this process. Whereas now if we imagine that methane oxidizing bacteria join the feast, right? They start to incorporate some of that methane, and as they become combined with your tissue, you might actually see a decrease, because again, those lighter isotopes are starting to get incorporated into your biomass. So what we did is we took our methane incubations before, right, like I showed you, with water and sediment, and we put some vegetation there. And then what we did is we measured the change in the isotopes of the vegetation, which is shown here, simultaneously while measuring the methane production rates. We left those bottles with oxygen available so that methane production and oxidation could be occurring sort of simultaneously. And what we found is a strong relationship between our net methane production rate here and the isotopes of that vegetation. And because that relationship is negative, we were assuming that that was because of this mechanism of the methane being produced and then oxidized and that getting incorporated into that vegetation, okay? Well, so we did that at a pond level. We instead took some vegetation, instead of putting it in a bottle, and we measured the change in the isotopes through time as it decomposed in a pond itself. Again, we find a strong relationship between methane production and the isotopes, but it's the opposite direction. We see a positive relationship here. And so that suggests that perhaps methane production is occurring at that sediment-water interface where that vegetation was placed, but we're not really seeing as much oxidation. And so we had to think about why that might be. And I guess the obvious conclusion is that a bottle is not a pond. Yes, so we were doing a lot trying to manipulate the chemistry and biology of these systems, but I think we forgot about the physics. And if you can imagine that a bottle here is capped to the atmosphere, right? You're getting methane being produced. At some point it might build up so much in this headspace here that it's going to diffuse back to where the lily or that vegetation was, right? And it can actually, that methane can be oxidized in proximity to where that vegetation is decomposing. So a simple way to think of it is like maybe we're artificially sort of forcing that oxidation to occur at that level. Whereas if we're thinking about placing vegetation in a pond, right? This pond is one, open to the atmosphere, so that methane is being emitted and is moving away. But the other thing too is that the wind can really stir it up. And so that's going to increase that, as well as you're likely to have more oxidation, but probably in the water column instead of right at that sediment interface. And so I think the interesting thing is clearly there's a difference based on the ecological scale of our experiment, but those strong relationships between the isotopes and methane cycling suggest that this could be a useful tool for thinking about these processes in the future. So now, for the best part of all, we're going to talk about methane production and how it's affected by climate change. The Copper River Delta is an ideal place for thinking about climate change. And that's because it's in close proximity to the ocean. And after the Great Alaska Earthquake, the delta is now subsiding, so it's sinking. So essentially, it's going to be at a risk of higher relative sea level rise relative to other locations. The other thing, too, is that northern latitudes are warming disproportionately. So over the last 50 years, temperatures in this part of Alaska have increased by about 2 degrees Celsius or about 4 degrees Fahrenheit over annually, essentially. And if you look at the winter temperatures alone, that's an increase of 3.5 degrees Celsius or about 7 degrees Fahrenheit. So that's a lot of warming we're talking about. And that's essentially changing the length of the growing season. And so we wanted to think about how some of these climate change mechanisms are going to actually affect methane production. We already know that warming is likely to affect methane production, increasing it, right? It's increasing those microbial processing rates from other studies as well as what I showed you earlier. But we know less about how longer growing seasons and sea level rise would affect methane production. So longer growing seasons means that the plants are going to have a longer time to grow. There's going to be more organic matter available for decomposition. As things decompose, that oxygen is going to get depleted, right? It's going to be used up by other organisms. And then fermentation is going to take place. And those fermentative bacteria make acetate and hydrogen, both of which are electron donors or food for the methanogens. So we thought that increasing the amount of organic matter for decomposition would also increase the amount of methane being produced. Now let's look at sea level rise. Well, it turns out that seawater tends to have a large amount of sulfate, and sulfate-reducing bacteria will compete for some of those same acetate and hydrogen, those electron donors that our methanogens, or those guys who are producing the methane, are going to want to eat or use. And so sulfate, remember, is higher on our tower of power than the carbon that our methanogens are using. So it's likely that they're going to outcompete our methanogens, thus decreasing the amount of methane that is produced. And so we first wanted to look at that organic matter part of the question. And what we did is we took our methane incubations, again with that sediment and water in a bottle, and we added some vegetation to it. We had some bottles that had no vegetation, those were the controls. We also added the most predominant macrophyte or vegetation species on the delta. There's this buckbean here, horsetail, lily, and then this is mare's tail. And so we added those to the bottles and replicated those across 9 ponds. And this is what our results look like. So methane production is on our y-axis here, and then here's our control, our buckbean, our horsetail, our lily, and our mare's tail. And so what you see here is that horsetail and lily really elevated the amount of methane that was being produced, whereas buckbean and mare's tail didn't as much. So that suggests that there's some sort of species effect here. And a lot of times, researchers have talked about the amount of nutrients or lipids in the tissue really matter. But the more we did research about this, the more we started diving into the plant or the botany research, we found out that buckbean and mare's tail tend to have antimicrobial compounds, whereas the other ones didn't. And so we think it's possible that those antimicrobial compounds were actually having a negative effect on those fermentative bacteria that produce the food that the methanogens use. So those antimicrobial compounds could have been sort of superseding the benefit of having more organic matter. So let's talk about the second effect of climate change, and that's sea level rise. And so what we did to think about this is first we wanted to do more of a long-term study where we looked at what kind of methane is being produced in our freshwater ponds versus our brackish marsh. That's our salty marsh, essentially. And so we compared those two production rates. And then we did an experiment where we took our freshwater pond sediment and we flooded that with the brackish or the salt water. And so that was sort of to simulate our sea level rise, so to speak. And so what we found is that methane production was much higher in our freshwater systems than in our brackish systems. And so we started to ask why that might be. And it turns out that sulfate in the sediments of the brackish system is much higher. And they also have much higher abundances of those sulfate-reducing bacteria, whereas the freshwater systems had lower sulfate and higher amounts of methanogens, or the organisms that produce methane. And so again, sulfate-reducing bacteria are higher up on that tower of power than methanogens, and so what happens is those sulfate-reducing bacteria outcompete our methanogens, resulting in decreased methane production. But interestingly, when we looked at simulating sea level rise, remember we took those freshwater sediments and we incubated them with water from their own systems, fresh water, and then we took them and we also flooded them with brackish water. We expected that methane production would decrease over here, but that wasn't the case. They were not different. But if we think back to our microbial results, where we had really high amounts of methanogens there, not very much sulfate-reducing bacteria, it just goes to show that perhaps the 14-day incubations wasn't really long enough to get turnover in these really slow-growing microbial communities. So let's come back to our conceptual diagram here to really wrap up what we've learned. We found that increasing organic matter tends to enhance methane production, but it really matters what species or the quality of that vegetation. As far as sea level rise, I mean, at least in the long term, it looks like methane production would be decreased because there's higher sulfate availability and higher sulfate producing bacteria. We didn't see the same thing in those short-term 14-day incubations. And so that kind of leaves us with the question, over what time period would we actually see a shift in the microbial community such that sulfate-reducing bacteria outcompete the methanogens? Another thing to think about too is at some point, if we have more organic matter for decomposition from the longer growing seasons, there might be enough electron donors to go around for everybody. So we're really not sure how these two climate change mechanisms are going to interact to affect methane production in the future. So we have taken multiple trips up and down this tower of power, and we've walked through and talked about how physics, chemistry, and biology really shape pond ecosystem function. And so wetlands are these amazing biogeochemical hotspots, right, where physics, chemistry, biology is really important, especially those microbial organisms, right? And I hope I've convinced you that microbial organisms, even though we can't see them, that we have to appreciate the contribution that they make in these systems. They really are the ones doing all the work. And I also hope that I've convinced you that the microbial warfare in this system via the Tower of Power is as exciting as Roman gladiatorial combat, and that we should all spend time thinking about the bottom of the Tower of Power, especially with regards to methane. And then lastly, studying relatively unimpacted systems like the Copper River Delta, where there are low nitrate levels, really matter. We saw how nitrate really had a dramatic effect on those biofilm communities. And that just goes to show you, like, how have we as human beings affected other ecosystem processes? And so I think if we want to think about restoring wetlands or systems, we have to know what they were like before human beings were there and had an impact. And with that, I would like to acknowledge some funding sources. Gordy Reeves from the Pacific Northwest Research Station really provided a whole lot of funding and advice. I like to call him the godfather of the Copper River Delta research. I would like to thank my co-advisors, Gary Lamberti and Stuart Jones. Gary's been with me since the beginning, and he has always put up with my stubbornness and allowed me to pursue things independently beyond both of our expertise, and so I really appreciate the freedom to do that. Stuart Jones, he, for whatever reason, whenever I was exploring something new, it just turned out that somebody in Stuart's lab was working on it. And so it just made sense to have so many great collaborations with him as well as his graduate students. And so that's been really great. I appreciate all of the inspiration that has come from the ideas with our lab meetings. Dom Challoner, he's been really fantastic. Because of his experience in the Copper River Delta, he especially played a really critical role early on, advising me, getting me started up there. And so I appreciated that, as well as being able to nerd out about isotopes. And we've talked a whole lot about intellectual virtues, a whole bunch, and that's really inspired me in how I conduct my science as well as how I teach. And so I thank you for that. That's been wonderful. Gin Tank is always encouraging me to look at the finer details, and I'm so happy that there's someone else who loves biogeochemistry as much as I do. So it has been a pleasure to have her. Adrienne Rocha is wonderful, always is asking me the big picture questions about why my research matters and getting me to think along broader context. I appreciate that. I recently was able to go up and work with some of his students up on the North Slope of Alaska, and that's a different type of rugged than I was used to. It also, I also learned that plucking tussocks is far more tedious than some of the chemistry work that I do. So I appreciated that perspective. The folks over at STUS have been amazing. I wouldn't have been able to come up with all this crazy chemistry data without them. Mike Brizicki, the Lamberti lab manager, is amazing, has always pitched in with lab analyses and just being an incredible resource. Some of the students I've worked with and I've mentored, Julia Hart, Melanie Runkle, Josephine Chow, Dana Smith-Evans, have helped with either field work or also lab work and have just been such wonderful inspirations. The Forest Service at Cordova, I really, with all those folks listed up there, they're all amazing. I couldn't have done this. I mean, they're the ones operating the airboats to get us out to these sites. There's the ones that provide the gun bearers to make sure that we don't get attacked by bears. They're really fantastic and with amazing attitudes. And their knowledge of the landscape as managers is just really fantastic and is really helpful. I'd also like to thank both the Jones and Lamberti labs. Will West and Jake Zort, both of them were particularly huge collaborators with me in my dissertation. Will West taught me everything I know about methane sampling, and Jake taught me a lot about ecosystem metabolism, and I'm very thankful. I'd also like to thank the Jones Lab. I gave this talk and they encouraged me to be really nerdy about the chemistry, and especially Dave Armitage. He gave me the idea of the Tower of Power, so I hope you all enjoyed that. And then the Lamberti Lab, Matt Cooper, Patrick Shirey, Nate Evans, everyone has been really fantastic. And so I just want to thank all of you so much for supporting me, friends and colleagues. It's been wonderful. And so I'd like to thank everyone who's helped along the way, my family for coming from Texas and Colorado to come see me here today. That's a picture of me and my brothers. I used to have long blonde hair. And also, I wanted to give a shout out particularly to Julia Hart. I know I mentioned her earlier, but she recently got her master's. She beat me on, studying, so she's really phenomenal. I look forward to seeing how her career develops. She also is the one that I worked with for my 4th chapter with the isotopes. And I can't imagine any undergraduate being as tenacious wanting to take on isotopes for their first project, so that's really impressive. And then last and certainly not least, I would like to thank my spouse, Mike Steffen. He moved to South Bend for me from Baltimore and has lived here ever since. And he has been an amazing support. He always encourages me. And I couldn't have a better partner or someone who is more thoughtful. And I really appreciate it. So I'm going to take questions now before I do it.